human biology students. I'm going to be talking you through the last section of Unit 1, and I'm going to start with the DNA and gene expression and how transcription and translation are enacted to make us have proteins, which are basically run the body. Okay, so I will be showing you a presentation. Okay, and I will share the screen with you now. Okay, so hopefully you should be able to see this presentation. Okay, so we will move on through, and let's hopefully we'll be able to work through it. So it's slightly complicated, but obviously as a video you can stop and go over it and make your own notes as you feel fit. Okay, so we've got our basic structure of DNA, which we had from when we did the first section on Unit 1, which is about the cellular structure, the ultrastructure of the cells, and we talked about DNA. And so DNA becomes chromosomes. The double helix is wound up, it's coiled up very tightly. We have special proteins called histones, which the DNA coils around, and eventually we end up with supercoils, and we end up with our chromosome. Okay, so inside the DNA, what those nucleotides are made of is nucleic acids, which make up the DNA and the RNA, and they've got three things that make them up. Okay, so we have a six-membered ring, okay, which contains nitrogen, a pentose sugar, which pentose, pent means five, it's a five-membered ring, carbon, 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 and oxygen at the top. Okay, and a phosphate group, so that's phosphorus is in there, and these parts make up the backbone of the DNA. And this is the key thing: is these nitrogen-containing base, which we can have several varieties of. So in DNA, it's the D stands for deoxy. Okay, so deoxyribose nucleic acid, whereas RNA is ribonucleic or nucleic acid. The sugar is ribose. Okay, so here are these nitrogen-containing bases. You can see they've got nitrogen here. So it's an organic chemical, six-membered ring, two nitrogens in the ring here, two in the ring here. But notice that this one has an extra five-membered ring attached to it. So the pyrimidines are all six-membered rings with some side groups, and the purines are six plus a five attached at the end. So you can see the groups there. Now, the way they've been drawn, we've got adenine and thymine. They match together, and you can see that the diagram shows that they sort of slot together. You know, obviously the adenine has got this arrow shape which fits inside this thymine, and the cytosine has got this sort of circular crescent shape which the guanine fits into. Okay, and what actually happens is that actually they make what are called hydrogen bonds. So they've got hydrogens and oxygens. Hydrogen is slightly positive, oxygen is slightly negative, and that sticks them together. So you've got two hydrogens in a hydrogen in position here, hydrogen in position here, an oxygen there, an oxygen there. I mean, it doesn't matter which side is which, but we have two hydrogen bonds between the adenine and the thymine. And so they will only pair up that way because they make two. But with the nucleotides guanine and cytosine, they make three hydrogen bonds. Okay, so that's why they would stick together. Okay, so that's how our double helix comes about in DNA. So the DNA structure looks a bit like this. So we can end up with a series of base pairs. So this code here, which is cytosine, A for adenine, G for guanine, T for thymine, they will pair up to match, okay, to make the DNA. And the sugar phosphate bit is this backbone shown here. Okay, so we have in effect what we call a DNA ladder. Okay, and they're anti-parallel DNA strains because, you know, they match up opposite each other. Yeah, so we end up with our double helix. I'll put a link to this video. It comes up, what does it say? It's a YouTube clip, so hopefully that YouTube clip is still working. I'll put a link up, and it will come up in the video here. Okay, so we get semi-conservative replication, okay, going on. So as the DNA opens up, we can match up the base pairs with each other to make a copy. So it's semi-conservative because this is the opposite to this one. Okay, and this is the opposite. The red one is the opposite to the blue one. Okay, so here, this is what's happening here. Okay, so we've got a point where the DNA opens. Okay, and then we have a leading strand. This strand is made slightly ahead of the other one. Okay, and the new nucleotides are being added on from this side, so going on there, and the lagging strand, the nucleotides are coming in this way, okay, and building up that way. And we have a protein called DNA polymerase, which enables these new nucleotides to come on here. So these nucleotides are free in the nucleus. Okay, they will floating around, and then they will come along. The DNA polymerase will stick them together. The helicase, okay, so helicase from helix, yeah, this is what's opening up the helix so that the DNA polymerase can come along and make the new, or add the new nucleotides on following the strand. So one of the gyrase unwinds it, so it's unwinded. The helicase unzips it. Okay, the free nucleotides are added to the exposed bases. So here's this one's exposed, this one's exposed. Okay, so the free ones are being added. Okay, the polymerase catalyzes the action of the new in the 5 to 3' direction. So yeah, so there is what we can say that the DNA has a certain direction. Okay, 5 and 3' ends. Okay, and the leading strand is synthesized continuously. Okay, and the lagging strand is joined later, so it lags because it has to be synthesized in parts. Okay, and there's videos on that which are animated, which are quite useful. Okay, so the central dogma came about from Francis and Crick, and they had help to actually get the structure with the X-ray crystallography. And so the DNA is like this. Okay, it's transcribed. Okay, so we can make mRNA from it. Okay, so we make mRNA from it. Okay, and then that is translated into our proteins. Okay, so the mRNA will come out of the nucleus, and in the endoplasmic reticulum, the rough endoplasmic reticulum, it will be translated into the proteins, which are packaged up and then pushed around the cell. Okay, so we can see it here in an overview. Here is the DNA. Okay, it's been opened up. Okay, and we're going to make the protein insulin. Okay, this section of the DNA codes for the insulin. So we make a messenger RNA molecule here that comes out into the Golgi apparatus. The Golgi apparatus has got the ribosomes on it. The ribosomes will then translate the messenger RNA. They'll read it, and they will make the protein inside the endoplasmic reticulum. As the protein gets made, the endoplasmic reticulum is like a series of membranes that will bud off. Okay, and then it can be made, pushed to the Golgi apparatus. It can be changed there if need be, but the Golgi apparatus is again another series of membranes. It's almost, a continuous series of membranes coming from the nuclear envelope, the outside of the nucleus. But this can be changed and modified, and then it's pushed out towards the membrane of the cell, and that will release insulin into the pancreas, and therefore out to the bloodstream. So this is going to, for the insulin, this is going to be happening in the beta cells of the pancreas. Okay, so that's protein synthesis, and we have a nice little animation of this. So hopefully this animation will open up. Okay, and I can put it over here, and you can see this animation. So many of the cells are involved in protein, which is central to all life processes. Click play. Okay, so nucleus, okay, consists of genetic material, okay, bound in the nuclear membrane. It contains one or more dense areas called nucleoli even. So transcription, here's the transcription happening. So we open up the DNA. Hang on, there's a U there. What's that? Well, U replaces thymine, RNA. Okay, so RNA doesn't have T, it has thymine. So you can see we've got a leading strand here. This one has to be made up in fragments. This one's continuously made, but we've got a lagging one because they just can't get in there at the same stage to actually make each other at the same time. Okay, so let's move through. So the ribosomal RNA, okay, is transcribed in the nucleus. This makes ribosomes, okay. Basically, the RNA folds up, or ribosomal RNA folds up to make the ribosome, okay. And then those come out of the nucleus and they get attached to the rough endoplasmic reticulum. So coming out of the nucleus. Okay, you can see that the endoplasmic reticulum is continuous with the membrane of the nucleus. So the ribosomes are stuck on the outside. Smooth ER has no ribosomes, hence it's smooth. The ribosomes make the rough endoplasmic reticulum rough, but it can store carbohydrates, lipids, and other molecules which might be added to proteins in the future. Yeah, so after the proteins are made in the rough endoplasmic reticulum, they pass through almost in a continuous manner to the smooth, where anything else is added to them. Excuse me. So eventually, you're packaging up all these proteins which may have been modified. Okay, and they package up into little vesicles or packets of proteins and the other molecules, the carbohydrates and lipids, etc., and they're transported to the Golgi apparatus. So they end up going to the Golgi apparatus, the membranes fuse, now in the Golgi apparatus, and this will modify those proteins, perhaps add the carbohydrates, the lipids, okay, and the vesicles are fusing with these are called cisternae, these folds, okay, fuse at one end, and new ones bud off the other end. Okay, so some Golgi vesicles are lysosomes, so these contain enzymes that can break down dead stuff, bad bacteria. Others are going to transport proteins to elsewhere in the cell or eject them from the cell through the membrane, and that's called exocytosis. Okay, so endocytosis is when the package from the Golgi apparatus goes inside the cytoplasm. Exo is when it comes out through the membrane. Okay, so that is it for that. Okay, so we'll move on. So that's like a real overview. So then we've got this genetic code. Okay, so this code is made up of all these bases. Okay, and we're going to go through the main features of the genetic code. So let's bring that up. Okay, so start. Okay, so match the features of the genetic code to the definitions. So I'm going to pause a little bit. You can think about how you'd match these up. It might be nice to draw a diagram where you link these together. Okay, so match the features of the genetic code. So I'm going to go through it now. Match the features of the genetic code to their definitions. The code is degenerate. Okay, it's non-overlapping. It's almost universal. It's a triplet code. So right, so match the features. It is almost universal. Given them it may be coded for one than the same signal bases code for the same amino acid in all organisms. So that is that one. Hang on, am I going to be able to move it? Can I move these? Why can't I move these? Okay, yeah, those two link together. Okay, it's a triplet code, i.e. 3 nucleotide bases, a codon codes for one specific amino acid. So triplet code is that. The code is degenerate. That means that actually you can actually map a 3-letter code to more than one an amino acid, but more than one 3-letter code could code for the same amino acid. A given amino acid may be coded for by more than one codon, and it's non-overlapping. Each base is only part of one codon, and each codon codes for one amino acid. Okay, there we go. All right. Okay, so that's obviously you can stop and check you've got your answers right. Okay, so let's move on. Okay, yeah. This is from BoardWorks. Okay, what's going on here? The writing's over the top. I might need to just very quickly move that writing. Let's see if I can do that now. What I'm on is chromosomes. Let's make that. Right, I'll just make that a bit bold. Let's just making this bold so it's visible. There you are. Okay, that's better. You can read it now. What are homologous chromosomes? So different organisms, different numbers of chromosomes. Humans got 46. This is diploid. Okay, chromosomes grouped in pairs called homologous. In each pair, one chromosome has come from the mother and one from the father. Okay, so in effect, we have 23 chromosomes, but then are paired up for mother's 23 and the father's 23. Okay, but it's not quite as simple as that. So we've got 23 pairs, yeah, okay, right, okay, let's just move that up there. So what is the genetic code? Is a sequence of bases in the DNA. It contains thousands of sections called genes, okay, all polypeptides are made from amino acids, so the sequence must code for the amino acids. So we have 3 in a row. CGT is one amino acid, ACT might be another, GCA another, GAT a different one. Okay, so you might have thousands of these and they make up one gene which might code for one specific protein. Okay, it's the same sequence of bases and codes for the same amino acid in all organisms. So that must mean that the genetic code evolved quite at the start of beginning of life, or really close to it. In fact, there's a lot of discussion about the fact that you know, DNA and RNA may have been the first molecules to be life. The genetic code right, so we do have DNA in mitochondria, and plants have it in their chloroplasts. They have their own DNA, and it's different to this sort of DNA. This DNA has come from mothers and fathers. The DNA in mitochondria has just been passed down through the female side. The size of genes can vary quite a lot. Okay, average genes got 10,000 to 15,000 bases. Okay, so that's 10,000 of these. Okay, paired up, obviously. Okay. So quite a lot of genes. Okay, so let's move, or average genes got a lot of stuff in it. So let's just, okay, so I managed to put this as widescreen so it seems better for you to see. But of course that means that it has messed it up. I thought I checked it, but let's push this up to here. Come on, stay. You're supposed to stay there. There we go, the triplet code. Okay, so this is going through the board work stuff. One basic was one amino acid. Okay, then the possible amino acids are only 4 because we've only got 4 amino acids. If 2 bases would equal 1 amino acid, if we had like GC was 1 amino acid, then we get 4 by 4, 16. Okay, if 3 bases, okay, can represent amino acid, that means we've got 64 possible amino acids. Okay, and that's what the situation is. We've got 64 possibles. And Francis Crick and his colleagues confirmed this in 1961. And there are 64, which means that amino acids can be coded for by more than one of the codes, because there's 20 essential ones that we need. Okay, so let's grab this and put this up where it should be. Okay, what is mRNA? So the triplet code of the gene is converted to messenger RNA. Okay, so it's transcription, which we sort of start to look at. It's a nucleic acid. It differs in that it's single-stranded, not double-stranded, and it's ribose, not deoxyribose. And also, instead of thymine, so instead of having a T, okay, it will have a U. So T will produce, the opposite code to T is an A, but the opposite code to A in DNA is T, but in RNA is U, uracil. Okay, so there's a slight difference in the mRNA. Okay, so when gene expression happens, it's switched on, it's expressed, it then gets transcribed into an mRNA strand, which is then translated into protein. And therefore proteins, enzymes can control this reaction, and it's going to control everything. And each of your cells has got all the genes for everything that makes you up, but not all of those are expressed at the same time, and they might be expressed at different points in your life. Like, for instance, when you're growing up and going through puberty and all that sort of stuff. So they're expressed at different times. Okay, so we're putting these amino acids together to make our primary structure, and that's coded for by the DNA and the RNA. So transcription of the gene is when we have got the mRNA strand formed. Translation is when the mRNA via a ribosome makes the protein or the polypeptide chain. Again, there's a video of this, which I'll put in the link. I'll link that up and it should appear on the video about now. Okay, so transcription. And again, I need to very quickly move that up. Yeah, there we go, transcription and codons. During transcription, the mRNA is built up by complementary base pairing. So, okay, so what codons in the mRNA transcribe from this sequence? So, right, see if you could stop the video now, see if you can work out what nucleotides I'm going to get in my mRNA. Remember, they're going to be the complementary ones. Remember that A pairs with T, T pairs with A pairs with T, but in mRNA it makes uracil instead, so A will make U. C pairs with G, and G pairs with C. So we should end up, if you worked it out, you can check it now, it should come out as that. So each base is only part of one triplet codon, and you see how when we've written it out, just for ease, we've made a little gap between each set of 3. Okay. And each of these will make one amino acid. So we get 1, 2, 3, 4 amino acids joined together from that code. Okay, so let's have a look at transcription. Okay, and we should have a nice animation of this. So here we go. Okay, so make sure it's reset. Okay, so a section of DNA is transcribed or copied into molecules of mRNA. Let's see how that happens. Firstly, helicase breaks the hydrogen bonds between the bases in a specific region, so that hits a specific region of the DNA for a specific gene, which therefore makes a specific protein. So strands separate. RNA polymerase binds to the base sequence. Okay, it's a promoter region. So let's just go back a second. So this promoter region might have a specific set of codes. So some of the, although we said that the triplet code is degenerate, i.e. the 64 possibilities, some of those possibilities are start codons, which tell the promoter to join on. So we start, we go from 5' to 3', okay? And it determines which way the polymerase faces and which region is used as the template, okay? So the RNA polymerase moves along the DNA strand. As it passes over it, it forms a complementary mRNA strand. And so they're sticking on, okay? Okay, but let's just recap on the direction. So it goes from 5, at the beginning you saw that this is going from 5' to 3'. So that little symbol there is pronounced prime, okay, and it's the direction of the DNA is running. Okay, so that one can go along 5' to 3', and we keep adding it, keeps continuously adding the nucleotides to make our mRNA. Okay, so we continue. Okay, 2 strands of DNA start to recoil behind it, so they're going to recoil up here, so it starts to recoil up. Okay, as that coils behind it. Yeah, until we get to an stop codon, terminator region, stop region, and that tells that to stop doing that, and the mRNA strand is released, ready for the next stage of protein synthesis, ready for translation. So on the other side of the strand, that will also be being made in a similar fashion, except that it has to go, still has to go 5' to 3'. So they're coming in this way, that's made in bits, so it's a bit more, that's why it lags. Okay, so that's the transcription. Okay, and there's a nice diagram of that. Okay, actually, I'm a bit short on that one, was nice. Let's go back a second. Transcription, if you want to have a look at that diagram, stop it, pause it, make sure you understand what's going on. Okay. Okay, so with a gene, okay, there are some bits of it, okay, which do not code for anything useful, okay? So when we've got the RNA that's been, before it actually is properly mRNA, we have to cut out the bits, the introns, which don't actually code for anything, okay? Because DNA has been made up from over millions and millions and years and years, and it has picked up loads of extra DNA in it. Some of it is completely useless, okay? So these are introns, okay, and they are cut out, okay, by splicing enzymes. So we then make our mRNA. Okay. So they're removed from the primary RNA. Okay, and they're joined together. Okay, so there are endonucleases involved in making, they splice it, so cut it up. So now we're on to translation. Okay, occurs in the cytoplasm, sequence of amino acids. Amino acids assembled into polypeptides. So amino acids are present in the cytoplasm. There's loads of them, and we need to use another RNA molecule. So the RNA molecule is tRNA, translation RNA, and this or tRNA, and it will come along, and on one end of the tRNA there will be an amino acid. On the other end of the tRNA will be a 3-base pair unit. So this is how these three ACG will code for a specific amino acid, because on the tRNA will be the opposite code to ACG, so UGC. Okay, so that will therefore carry this amino acid. So we started from this direction here. This one's already been made. This one's already been made. This one's been made. This one has just come off of this tRNA molecule. This tRNA molecule is coming away. It will go back into cytoplasm and pick up some more of the specific amino acid, well, this green one that it was. This one has been connected and is getting ready to break off from the tRNA. And then this one is just coming in, and this one, it will then make a peptide bond to link the two amino acids together, and so we'll end up with our polypeptide chain getting longer and longer until we get to the end, until the stop is reached. Okay, so let's just adjust this very marginally. So what is translation? Once it's been transcribed, it moves through a nuclear pore, combines with the ribosome. So there's the ribosome. That's what is reading the code. Okay, and we get the correct amino acid transported via tRNA molecules. So this is a close-up of the tRNA molecule. There we go. Okay, so the amino acids attached to transfer or translational transfer RNA molecules specific for one amino acid. Okay, so the amino acid is attached at the top, okay, and at the bottom, yeah, so these, this is all nucleotides, like the RNA, but it's the three at the bottom here, which are an anticodon. They are the opposite to what is on the messenger RNA, or complementary, yeah. So what is the anticodon for the codon AUG? See if you can work it out. You should have got UAC, okay, so on the RNA, the messenger RNA, there will be UAC, and this codon here could be AUG, and it will stick. So let's just move this up a bit. So what happens during translation? tRNA will attach. So the ribosome is making this specific bit available, and this tRNA comes along, and this is the amino acid methionine, and this is going to come along and be attached. Okay, and once it's done that, the tRNA is released. Okay, and then it can go and pick up more methionine. Okay, so then the ribosome is going to move along. Okay, and we then get, in this case we've got cysteine, and then is that asparagine or aspartame? But there's a 3-letter code for all these amino acids. I think that is asparagine. Yeah. Okay. Then we're building up this polypeptide, and eventually it'll be completed. So let's watch this in action. Okay, so here's the animation. Okay, just check right at the beginning. Right, so translation, formation of the polypeptide from the mRNA base sequence. Each codon codes for one amino acid. So the mRNA strand is formed, the ribosome comes along. At the start, in this case AUG is probably quite a common start codon. So that gets attached, okay, and then transfer RNA comes along, okay, with the anticodon, which matches this one. Okay, it's got our methionine on, okay, and then another one comes along, the ribosome moves along. Yeah, this one is leucine. Okay, we make a polypeptide bond. Okay, then the next one will come along, it's probably threonine, I think. That the one that had the methionine on is now detached. It doesn't have methionine connected anymore. It can go and pick up some more methionine. Okay, so keep going along. Yeah, more serine. And then we get to a stop codon. Okay, so this is a special sequence to stop codon, and that will then mean that it comes off of the messenger RNA. The tRNAs are returned to the cytoplasm to pick up more copies of the amino acids, and now we have our, in this case, rather short section of polypeptide chain with 6 amino acids on it, and it's released. And it can go, well, it will undergo post-translational modification. So the polypeptide chain, if it's longer, would then fold up and make secondary and tertiary and potentially quaternary structure of the protein. Okay, so that is that. I will move that out of the way. Okay, so let's see if we can work this out. I think this is going to take us all the way from DNA to the amino acids. Okay, let's just check and this is just a little checky thing. So anticodons on, well, let's start with the base triplets on DNA. So if we have A, okay, it's going to be paired up with, okay, well, it's going to be T. Okay, in fact, if I let you pause the video, you can match up. Oh, hang on, this is, basic, the codons on the mRNA. So if that's G, that one is C. If this is T, on here, T pairs with A. Okay, A will pair with T, G pairs with C, G must be C, and right, so now we need to work backwards. This is G, so this must be C, so this must also be G. Okay, so if this is C, that must be G, and therefore this must be C. Okay, so GC, and that will make G on that one, on the tRNA. So C, that's going to be a G on the tRNA. Okay, so that one's done. So U and A is T. That's all done. So we had a C here. This is going to make a G on the mRNA, which is going to make a C on the tRNA. So we A here, that's going to make a U. Okay, because remember with the RNA, we don't pair with thymine, we pair with uracil. Uracil will pair with adenine there. So let's just check and hopefully got them all right. Ready, 3, 2, 1, bang. Good job. Right. Okay. So right, amino acids. So right. Okay. Now there is this table which we can use to work out which amino acid is which. So this one, so what we're looking at, okay, so we've got A, C, C. So A, C, and C should be threonine. Okay, so we've got TTG. So TTG, so the first base is, right, we need to work from the, we need to go back and work from the, from the actual messenger RNA. So this one is AAC, so A, C, so that one should be that. We need to go back and do that one again then, don't we? So we've got UGG, so U, G, and G. Let's try that, okay? And this one, okay, so hopefully it's from the mRNA to the amino acid rather than tRNA to the amino acid. Okay, so we've got CCG, so C, and G is proline. Okay, let's check that. Nice. Okay, so yeah, so it's the opposite to the DNA. It's the code is opposite to the DNA. So it's this one, UGG, is going to be tryptophan. Asparagine, proline. Okay, good job, and that's it. So what's S do? Can't remember. Anyway, that's it, done. Okay, so right, so reading the code, we can use this Okay, so the first position on the now this is starting from the DNA because we've got a T here. Yeah, so we can start from here, and you can see that we can work out what is the first position, what is the second position, what's the third position, and then we should end up with the correct thing. So we could have something like TAA, and that's a stop codon. Okay, so that would stop the polypeptide synthesis. We can also use this wheel here that can do it. So if I had GGG, I've got glycine, or GGA is also glycine, or GGC is glycine, or GGU is glycine. Okay, so you can see how we've got, so this will give us our, you can see how this gives us our 64. We've got 4 in the middle, 4 along here, and then 4 in each one of those. Okay, that will give us our the possibilities of all our amino acids. Okay, so let's have a look. So you can stop and try and work out what each of these is. Okay, and we'll go through the answers now. Okay, so stop before and then try and work out what AAC is. So we'll just do ACC together. So A, C, and C is going to be threonine. Oh, we haven't got the answers, have we? Let's have a look at the answers. So, yeah, so AAC will be threonine, GG, so G, okay, GGA is glycine, CUU is leucine, UAC is tyrosine, ACA is threonine again, ACG is also threonine, AAG is lysine. Okay, so you can see the degeneration, there's lots of codes can be threonine. Okay, right, so okay, that's all well and good, but we can have mutations. Okay, obviously a picture of the X-Men who are, obviously for mutations. So mutation is where we got a random change in the amount or arrangement of genetic material in a cell. So we can have DNA, we can change the nucleotide base of the sequence. We could have chromosome mutations where a whole chromosome is changed, or parts are changed, or cut out, etc. So that is chromosome mutations. So what can do that? A mutagen is a physical or chemical agent that can change genetic material, and thus increase the frequency. Now they would have that symbol, okay, and you can see it's sort of like, a star on a person with bits breaking off, which is well, yeah, looks nasty, doesn't it? But the things that are mutagenic, carcinogenic, or toxic, they can change the genetics, so that's going to affect reproduction as well, because obviously you pass on the DNA in reproduction. E.g., some examples: tobacco, UV light, X-rays, arsenic. Yeah, all of those things are going to be mutagens. So the effect on the primary structure. So the primary protein, we've got our sequence, our chain. The secondary structures, we've got beta pleated sheets or alpha helixes. Now, if one of these is a wrong amino acid, then it can change what happens here, and that then can have a knock-on effect on the tertiary structure, which can have a knock-on effect on the quaternary structure, where multiple polypeptide chains can come together. Okay. So, a point mutation is often silent. So, for instance, here we've got a sequence which produces the protein, the amino acid sequence: methionine, alanine, asparagine, GLN, serine, leucine, arginine, and then a stop codon which stops it. Now, with a point mutation, say we get that A here changing to a G, okay, then instead of getting well, this one is silent, yeah, basically because it's the codons are degenerate. CAG and CAA both it's glutamine, yeah, but anyway, they code for the same amino acid, so that's no problem. Okay, that's not going to cause a problem. Okay, no effect on the actual protein, so therefore no effect on the actual organism. Okay, so we might get missense. What about this point mutation? So we've got the same code again. What happens if we change that to a C rather, so T to a C? Okay, that then makes serine instead of leucine. So that's a different amino acid with a different R group, a different side chain, and that could therefore have an effect on the structure and the function of the resulting protein. Okay. Okay, so another point mutation nonsense. Okay, so what about this one? Okay, so here, right, we've got methionine, alanine, asparagine, glutamine, I think, and then serine. But instead, if we've got that C changed to an A, instead of making serine, we've got a stop codon, and that will stop the polypeptide chain there. And so we have a small bit of polypeptide chain, smaller than it should be, which is, could be absolute nonsense. Yeah. It won't be built fully, or it won't function properly. So that might mean a specific protein doesn't work in a certain organism. We could input an insertion mutation. Okay, so this is what would happen here. So here we've got AAC, but now we've added an extra nucleotide. So now we've got AAA, and that C was the same C here. So we've shifted them all along one, and we've got an extra one at the end, and that's completely changed some of the amino acids because we've got a different code on here and there, etc. So we change the red ones, we've changed completely. That's called a frame shift. We've moved the ribosome, where the ribosome gets its code for the amino acids has been moved along. So you can imagine the ribosome comes along, normally AAC makes asparagine, comes along here and it reads AAA and makes lysine instead. So it's all shifted a bit, and therefore many amino acids could be changed, which could be pretty disastrous. A deletion, okay, so that's the opposite of an insertion. So here we've got ATG, GCA, and the G has been lost. Okay, so again we've moved all the codons along, so we get different amino acids again. Okay, so this is it's also a frame shift. Okay, again, dramatic effect. Many different amino acids could be changed. The protein might not work at all, or it might do something disastrous. So mutations could be inherited or acquired. So if you have a mutagen, they're acquired. Okay, so something in the environment would cause your DNA to change. However, if you then pass that on, then the mutation's been inherited. Okay, and obviously at the moment we're looking at viruses, and the coronavirus that is picks up lots of mutations all along the way. So obviously it doesn't have parents, it's not necessarily considered a living thing, but it does have mRNA in the virus, and that gets changed, mutated. So if they're passed on, it's inherited, and they're going to be inherited in all the cells of the body. Acquired might only occur in certain areas of the body, like for instance the reproductive system. It's an acquired mutation then becomes hereditary or inherited if it gets passed down. Sometimes they're not passed down, so they would occur in somatic cells, so cells that are reproducing inside our body to repair stuff. So that means that then in that organism, the new cells, any new cells that are made are going to have the mutation, but if they're not in the sperm or egg cells, they're not going to be passed on. So some mutations occur spontaneously and randomly in the genes, and others are environmental. Okay, and so there is a summary video there, and this is it all done. Okay, so that is it, and I'll try and put links to the summary videos and the other videos in this video. So thanks for listening, and hopefully you can use this video to make sure you have a good understanding of this ready for the exam in May. And goodbye.